And then you learn. Welcome, welcome. Good evening. My BpharmMendelian weekend. Good evening. Just chat with me. Hi, hi, hi. Evelyn Ogada, hi. Today we are revising genetics. BpharmMendelian, hi, hi. Welcome on board. BpharmMendelian weekend or BpharmMendelian upgrading. All my students, you'll just say hi because the channel is yours, but today is the day for pharmacy weekend. Hi, Augustine. Hi, Edwin. Hi, Margaret. Hi, let's just say hi, BpharmMendelian. You just say BpharmMendelian and then we know we are together because I have many students, many kids. They're BMLS, they're MBCHB. So they might be wondering, why is Madame live yet she didn't tell us? So we communicated the BpharmMendelian, correct, welcome. Oh, perfect. I think we are totaling to 70. So we still give 5 minutes. We invite students and the class reps for both groups. Just tell me class rep group B. Hi, we are there. Kina Nick. The other lady also, please class reps. Yeah, Paila Ketuke. Hi, welcome. Yeah, Nick is there. I've seen Nick. Class rep group B. Class rep group B. Hi, everyone. Yeah, Nick, I know group A. Class rep group B. We are going to invite members for 5 minutes. And then we are going straight to the questions. I sent them yesterday. I can say in the morning, I think. But it's, oh, good. Class rep group A, welcome. Thank you so much, Jackie. If I've missed your name, I'm sorry, but I welcome all. Boaz Dulo, welcome. Kemoy, yes, welcome. So you can be visitors here. The way we do it, I send the questions and we'll always be referring to question number. Then I'll show on my small board, which you are seeing at a small corner in my house, how we are going to tackle the question. So in the charts, hello, Roslyn. In the charts, you are going to tell me your concerns, what you didn't get. So just by chatting, because I'll be able to read the charts. Moha, welcome. So I'll be able to read the charts, and then that's how I'm going to respond, because I don't know how YouTube can make it so that I can listen what the students say the other side, but then it's still, we can still manage with the charts. Huh? So I said we were to invite people 5 minutes. I can see we are at 41, which is fine. Essie, yes, hi. Janet, hi. So we are going to follow the questions the way we sent them, and we are going to cover so far the topics which we've done, the genes, the chromosomes, but basically Mendelian genetics will be taking the major part of tonight's discussion. And we'll see if we can tackle all questions. If we can't, I always say the world is not ending today. It is you people to tell me, Madam, which day before the cuts. Because on Wednesday, I have the group. You can see on the board, that is biomolecules. I had revision with them yesterday. So we didn't finish because, you know, students can be tired. Human beings can be tired. 1.5 hours was just too much. So we didn't do essays. So on Wednesday, I have these students to finish with the essays. So let's see how far we'll go. But I hope we'll be able to cover all the questions and the students will be able to understand. Yeah, Ebo Kati, welcome. Wambui, Lucy, Cecilia, sorry, Lucy. Bashir Mohamud, yeah, you are all welcome. So let me check my questions because I have to refer. I have to refer to my questions. And then we'll go straight. Hi, yes, Kialo. Kialo Tits. I think it's Titus. Kialo Tits. Samuel Ochieng, welcome. Class Rep Group B, yes. Agio Immaculate. Welcome, my dear. So we have all the questions. Class Rep. Class Rep Group B kept on reminding me, Madam, send the questions. But when she got it, she didn't even tell me that she has gotten the questions. Or you responded, it's me, I didn't see. Yeah, but it's good to say you got. Even Nick, I had to ask him. When he said, yeah, we got it. Yeah, yeah. So you just reply and say, oh, okay, we got it. So it's okay. But it's fine. If everyone has, let us just go straight and start as people keep on logging in. So you keep on liking, we subscribe. So in the channel, if you subscribe, you can easily join because you'll see when I'm live. I don't know the other way, but then you can also share with a friend. If a friend is already in the system, it's easy. We can move together as one. That is when we revise together. If you have a friend who is already subscribed, we can still share and be on the same page. So as we check in, yeah, the name is too long. Hello, I can just see entrepreneur. Welcome. So let me open the questions and we go straight to the questions because so far, whatever we've covered, if we discuss on how to answer those questions, then it can be of importance to the students so that you don't get worried at the point where you see a question and its applicability becomes a challenge. So revisions will always help. Whatever you did not get in class, you'll be able to get something. And I'm happy about the revisions on the channel because the students have been able to improve much in their performance. Because what if they didn't get to in class and maybe somebody do not want to ask or somebody feels that they can't ask, we go to the revision on the channel, you'll find that a student will understand more. So they'll be able to understand or be able to answer correctly when we get the questions. Keep on coming in. Elisa, hi. Edwin Karaoke, welcome, welcome. I'm just opening my questions. It takes a bit of time, but not that much, a few minutes. And then we go straight to each one of them. So I separated them by putting the Mendelian, the dihybrid, the monohybrid, the autosomal dominant, autosomal recessive, so that you can remember easily. Munene, welcome, welcome. Joy, welcome. Nyanchoka, welcome. Yeah. Welcome all. I'm just opening as we log in or as we check in, just say hi. Stanilas, hello. Welcome, welcome, welcome. BpharmMendelian, yeah. So let me open. Just give me a few minutes. I have to refer from my laptop because I'm projecting using the phone. So we are going straight to the questions because you'll find that some of the questions will repeat themselves in essays. Well, now we have to discuss in details. So we'll just go as per the questions. how they are asked and how you need to answer. So let me rub the yesterday's work so that it doesn't confuse you. You think you're in a wrong place. Not really. I just rub. I don't know what's happening. It's very slow. But I know it will happen. I'm still saying hi. Oh no. I already said hi to Nyanchoka. So keep on logging in. Oh, good. I've opened it. So we are starting with the Mendelian genetics. Any applications on Mendelian genetics, but basically, Mendelian genetics is wide. We have the principles of Mendel, the three which we learned. We have the monohybrid, we have the dihybrid, we have the Mendelian disorders. So it's a bit wide. So that's why I had to separate. We are talking about Mendelian genetics, but now monohybrid cross. So the first one says, in border collies, I said, as much as I don't set the same question, but applicability has to be the same. We have to understand the concept here. In border collies, black coat, B, there I'm even fair, I'm saying, is dominant over red coat. So you know very well the dominant allele will have the capital. And I said I have to include the letters, so that somebody not come with Z, somebody T, somebody Y, somebody U, uniformity. So in border collies, black coat, capital B, is dominant to red coat. A breeder has a black male who has won numerous awards, and the breeder would like to use the dog for breeding if he is a pure bred. It's even saying there, homozygous dominant. To learn this information, she test crosses. To learn this information, she test crosses him, that is the border collie, with a red female. Draw a Punnett square for F1 and F2 and state the genotype and phenotype of F1 and F2. So you are talking about the what? The coat color. Don't start and say we are talking about border collies. The coat color. Trait. Coat color. Alleles. I said all these are going to give us marks. Alleles. They've said capital B. Put it very well. Black, small b. For which one? For red. Correct. Look at the statement. They are saying, now let's look at the statement first. A breeder has a black male. A black male that has won numerous awards. A black male that has won numerous awards. And the breeder would like to use the dog for breeding if he is purebred. He would like to use the dog for breeding if he is purebred. To learn of this information, she test crosses him with a red female. She test crosses with a red female. They are talking about purebred. Look at that. So they are saying you have to give the genotypes for the F1 where this breed came from. Remember, black is dominant over red. So I told you, and we agreed, ladies, let the males be up here, females be up here. It means for us to get the F1s, they're talking about pure breed, pure breed. It means they will come all heterozygous. They will come hybrids from pure breeds. The parental generations, they have to be capital B, small b, small b. That is how we agreed, the pure breeds. 11 high, so it will bring the hybrids. It means the law of dominance of Mendel, still the allele for black will outride the allele for red. They will show they are black. From here, these are the F1s. First filial generation. They are saying from here, a test cross has to be done with who? Homozygous recessive. This is the male who has won award. The female, this is from the F1. So you are saying the breeder would like to use the dog, that male who has won. That male who has won numerous awards from pure breed, the parental generations. Now, to learn this information, she test crosses him with a red female. Red female. The red female, the recessive allele will only show in a homozygous recessive state. So, these are F2s. Second filial generation, they're asking. And I said there, draw a Punnett square for F1 and F2s. The Punnett square will add you marks. I think that marks is too little. 4 marks is too little. Because in those alleles, I always get half mark. You see, that allele and trait, you get half. And for me to balance it, I have to balance also just by answering the genotypes, because the Punnett square will get much of the marks. That is where you'll get the information. You'll know whether you are going to get the correct answer or not from the Punnett square. Now, just by stating the genotypes and the phenotypes of F1s and F2 generations. So for the genotypes, let's start with the genotypes for F1. You'll say all alike in brackets, capital B, small b. All heterozygous. So you'll talk about all alike, F1. And then the phenotype, all alike in bracket, black. Then you go to F2, that one you've talked about all alike in brackets. That's half mark. The other one half. Already those ones were half. It means the Punnett square, this question should have like 5 marks. The Punnett square should scoop at most 3 marks, at least 2. Because I'm looking at placing the male and female. Very important. Did you get the genotypes right? Did you get the information right? The parental generation or the genotypes of the parents. Hi, Derek. Welcome. Now, from the F1s you've seen, they will come out to be all hybrids. Test cross, I said, it is for their known genotypes. So you can do a test cross using the homozygous dominant or homozygous recessive. But in this case, they specifically said a test cross is done with this one who has just won awards. Look at whatever is going to happen there. 50-50. You have genotypic or the genotypes. They didn't talk about the genotypic ratio. The genotype, capital B, comma, small b, small b. Phenotypes, black, comma, red. If you are not asked about ratios, don't put ratios. They're asking about genotypes and the phenotypes. So for the F2, you can see we have both the heterozygous and homozygous recessive. So you have to put the genotypes there the way they are appearing, capital B, small b. Don't put in words. I said in words is not sure that they're genotypes. We only have the 3 possible genotypes. It's either homozygous dominant, homozygous recessive, or heterozygous. So in the F1s, F2s, you can see we have the heterozygous and the homozygous recessive. And then for the phenotypes, it's going to show black, comma, red. Straight to the point. Question number 2. If you have the questions, please just chat. I'll be able to see. And I'll be able to clarify. Let me just point this side so that you don't miss something. Yeah, we are going to manage. Don't worry. One day it will be a very big class with a very big machine. We can project and we can be heard. So don't worry. Step by step, we shall get it right. As long as the students can understand, I'm safe. So let's move to the second one. It says, I'll check on the charts. If you have a concern, just do the charts kindly. I'll answer all as much as it relates with our topic of today. If it's not off topic, the off topics are the ones which I'll assume. But the ones which relates with pharmacy today, genetics, I'll apply. I'll answer. A pea plant is the F1 offspring. Please project the question. No, I can't project the questions. I'm sorry. That is why I was giving the email. I can't project. That is why I asked you for. Please, I'm sorry. Question 1 for parental. For F2 phenotype, can it be 2 black, comma 2 red? It is still okay. As long as you put that comma, it will show that the phenotypes are black and red. That is Margaret. No problem. Even if you had for me too. Why should I give you a wrong? But you know, they're asking about a phenotype. And mostly whatever you are trying to express there goes with the ratios. So even if you put for me too, you are not wrong because you are describing for me the phenotypes which are present there. That comma adds more to it. It makes sense that they're the phenotype, not the phenotypic ratios. So that is correct. I was just referring to Evelyn. Evelyn, kindly check. One of your classmates can forward, but I think you can access the email, all of you. Clarify on the part of genotypes and the phenotypes from question 1. I said, Pilar. I said, Pilar, the question is asking, we draw a Punnett square, we already drawn. Huh? They are saying, if you look at the question, it was saying that this type of dog, is it a cat or a dog? I think it's a dog, comes from the parental generations which were purebred. If you talk about pure breeding, we have homozygous dominant, homozygous recessive. You can see it on the board to our left. From there, 1 out of these 4, where you can see their genotype is alike, is one of these dogs. So you are told to show from the pure breed of the parental generations, can you show the genotypes of the F1s? If they are pure breed, those parental generations, you are going to get all hybrids, all heterozygous. Meaning, we started with F1. They talk about the genotypes and the phenotype. The genotypes all alike, in bracket, capital B, small b. That is how I want you to write it. Still, the phenotype there, all alike, in bracket, black. F2, the genotypes, put capital B, small b, comma, small b, meaning put heterozygous, comma. We have heterozygous and homozygous recessive, meaning in the phenotype, you will have black, comma, red. So Margaret has also suggested that if she writes 2 black, comma, that comma tells about the phenotypes which are present there. It is not a colon. Colon talks about the ratios. So the comma will separate the 2 phenotypes. So if you talk about a phenotype, we said the genotype will control the phenotype. The genotype is the remote. Whatever you see out, there is a gene which controls it. So we talked about the genotypes. Whatever you are seeing here, we have the possible genotypes. It's either homozygous dominant, homozygous recessive, or heterozygous. And they are the ones which will tell the outer expression. But then this will follow the Mendelian law of dominance. In heterozygous state, it will still express the dominant allele. In homozygous dominant, it will still express the dominant allele. But now, Margaret has a lot of questions, right? Margaret, I don't understand your question. I was finishing to answer that up there. Then you say what? For F2 generation, I thought we still use the F1 hybrids for male. That is what is there. Margaret, the statement says we test cross using homozygous recessive, meaning the red. Am I the one who is not getting what Margaret is asking? Where did the female come heterozygous? In the statement it says, this dog who is a male, from the hybrid here, the heterozygous, is test crossed by, oh yes, just wait, I explained to Margaret, I'll be able to answer. We won't go to the next question until I answer. So, Margaret, the question says, from this male, you see, the heterozygous, it shows that it's black. From the F1s, a test cross was done with a female who was red. That is what you can see on your right. So where you come with the female, heterozygous, kindly, I don't understand. Maybe you can explain to me what you mean. Let me look at the second one. So when crossing the F2 for the female, you use the same as the mother. No. Arnold. Not really. Don't confuse these things. I said, when you are doing the test cross, many a times you will get the pure breeds from the parental generations. From that pure breed, if you cross any of the pure breeds, you will get the hybrid. And then from the hybrid who are heterozygous, I hope I'll be able to answer the other one because they're coming in. Correct. Joy is trying to explain whatever is there. So we explained that a test cross can be done using the homozygous dominant or homozygous recessive. And Joy is answering by saying, we have been instructed to use homozygous recessive from the question itself. And I said, Whenever test cross is conducted, it has to specify. You can't just leave it open to the students that a test cross was done using what? It's either homozygous recessive or homozygous dominant. So in this case, for us to see how the genotype of the black is represented in the next generation, which is F2, a test cross was done using the homozygous recessive. Because in F1, we could see as much as there were pure breeds, but all of them appear black. What if we get one, which is red, the recessive one? We do a test cross with homozygous recessive. Will we still have traces of this breed which will win the what? They talk about the dog won awards, and you can see half of it. Half of them will win. So the test cross we've been instructed, but the F2 generation should be gen F1. Of course, that heterozygous come from the F1, but now you've been instructed to do a test cross. AC Mudoni. Boaz is saying hi. Margaret is still saying, class, you had to say to find F2, you cross the F1 with each other. Did they say self-cross? Nothing like that. We are doing test cross. If you go, Margaret, if you go to the questions, the lectures, the main aim of the test cross is to identify the unknown. So in this case, you have been instructed whether you have to do a test cross from the F1s using homozygous dominant or homozygous recessive. Mutembe is saying hi. Malim is saying hello. Yes, so we'll be able to understand. Please, Margaret, don't mix things up. We'll be able to digest pole pole. But the point here is, they've said from the question very well, if you read it keenly, pure breeds, homozygous dominant, homozygous recessive. When they are crossed, you are going to generate. That is why the Punnett square is there. It's going to sort out all our genetics problem. So if we understand the equation well, the Punnett square will be right. So at that point, you can see all of them come as hybrids, heterozygous. Then from there, that is where you can see the heterozygous is up there. Do a test cross, correct. I'm happy that you've understood. Do a test cross using homozygous recessive. We are at question number 2. I'm happy. We are going to explain until we get, and that is why we are here. Yes. So a pea plant is the F1. Hi, Sam. A pea plant is the F1 offspring. Is the F1 offspring of a true breeding plant. You get? A true breeding plant with purple flowers and a true breeding plant with white flowers. It means pure breeds. Purple is crossed by white. Let's continue and see which one was dominant. This plant is crossed with one that has white flowers. You know? They already gone through that, the results for at F1, and then now they're asking at F2. So let's start. A pea plant is the F1. It means at the end of this live session, the video will be saved on the channel. So you can go anytime and listen to where you didn't get right. A pea plant is the F1 offspring of a true breeding plant with purple flowers and true breeding plant of white flowers. This plant is crossed with one that has white flowers. How many of the plants in the next generation will have white flowers? They're already asking at F2. So use P. It means capital P will stand for the purple, which is dominant. I think that my equation was a bit incomplete. P for purple. So you say flower color. What are we describing? color of the flower. Flower color. Alleles. I like writing with the black. It's a bit smooth. Alleles, capital P, purple. It resembles the ones which were in the assignments. Small p, white. Write it very well because these ones you'll score marks. When I see whatever you are describing there, I'll know the way to go. It will lead me to where I need to go. The outcome from the F1, we use it to generate the F2 using the recessive red. Okay. Pilar is trying to explain to Margaret, but Margaret said, okay, she has got it. It's okay. If somebody can also support or trying to explain to somebody, still okay. But now we can still proceed as we also keep on understanding. So we are talking about flower color. P is the dominant one, just by showing their capital P, purple. Sometimes somebody will add purple color. I'll still give you a mark, but it's a repetition because you already know the flower color. It's either purple or white. So they are saying they are pure breeds. True breeding, true. It resembles whatever we've just done for the dog. Punnett square solves a lot of problems. Genetic problems, that's what I mean. So it might look like a test cross, but it's not a test cross. It just directs us what to do. You have, it says, let's go back, what it was saying. True breeding. A pea plant is the F1 offspring. It's one of those hybrids which are there. F1. See, these are F1. These are parental generations. These are parent 1 and parent 2. The F1. So they're talking about a pea plant is the F1 offspring of the true breeding plant with purple flowers and true breeding plant with white flowers. So you can see the white flowers, the purple one. And they are talking about true breeds. Then this plant, they are talking about this plant, meaning it is one of these. This plant is crossed by what? They are saying that plant is crossed by white flowers, recessive. They're even saying white flowers, recessive. Yes. So it means, just put it very small so that we can see. That is too small. Still, let's assume this is a female. That is a male. So you have to put it there. So they are saying this plant is crossed with one that has white flowers. So you know the homozygous recessive, they will only be expressed when all the alleles for the recessive is present. So they only get expressed at homozygous recessive state. So we have the white flowers because white was represented by the small p. What is the question? How many of the plants in the next generation, that one here, F2, how many plants? Because they are talking about the next generation, because they're saying it is outcome of the F1s. They are saying the F1 offspring in the next generation, in F2, will have white flowers. In that F2, how many? Did they talk about how many? How many of the plants? If you talk about 2, because it's 2 out of 4, or you can talk about half, or 50% of them will turn to be white. Either or, it still expresses, yes, 50% white. You are still correct. Why should I give you a wrong? 50%, correct. Somebody has a question there? Question number 3? as the question directs. Just answer the question the way it's needed. Don't waste more time because you have the other questions to answer, but try to internalize, digest, understand, put it in writing. Punnett square is going to solve all those problems. And if you understand the question well and you draw the Punnett square, you are going to scoop an A in genetics. You will understand why is Madam now setting this. Question number 3. Ability to curl your tongue up on the sides, which is known as tongue rolling, is dominant, capital T. Let's go with the story. It is dominant to not being able to roll a tongue. We have to start with the trait. Tongue rolling. Tongue rolling. Tongue rolling. Something which describes precisely what you are talking about. Tongue rolling. Trait. Tongue rolling. Let's finish with the alleles. They talk about capital T rolling. Small t, not rolling, not rolling. Not rolling. We've clearly written the traits. Now we go back to our question. A woman who can roll her tongue marries a man who cannot. Look at that. Let us not rush whether it is homozygous dominant or heterozygous for this woman. For the man, you know, the man cannot roll. Let us do the Punnett square before we can see what they are saying. The man will put them there up. That's how we agreed. The woman will come here. Let's listen to the story. It says, a woman who can roll her tongue marries a man who cannot. Obviously, this man is agreed. Correct? That man has to be small t, small t. That is obvious. And we are saying there is a story for us to tell much about the genotypes of this woman. It says, a woman who can roll her tongue marries a man who cannot. Their first child has his father's phenotype. Obvious. It will tell you that the woman is heterozygous. How will the child come out to have TT, small t, small t, if the woman is not heterozygous? At the point of segregation, where the gametes form, the woman has to contribute the small t, and then the man will give it small t. But if the woman is capital T, homozygous dominant, then we'll have all hybrids. All the children will be able to roll their tongue. But there is one who can't roll because he has the genotype for the father. So, obviously, the woman is heterozygous. Let's see the questions. If you draw the Punnett square like that, I'll give you mark. Even if I don't say if I don't explain that you draw the Punnett square, you have to draw the Punnett square. It is going to give us marks. So what are the genotypes of the mother, the father, and the child? Simple. You already have it here. The father, homozygous, recessive. The mother, so don't tick. Somebody will say, Madam, you ticked. Mother, father, child. Full stop. You get your marks. If the woman can roll the tongue, won't it be capital T, capital T? Nyanchoka. I want you to write in your paper where you are. If you put capital T for the woman, what will happen? I want you to answer me there. Because where you are going to be caught is that one of their children has the genotype of the father, cannot roll. So if you put capital T, Nyanchoka, we are going to get heterozygous, all of them. And the law of dominance says the dominant allele for rolling of a tongue will outride the recessive allele for not rolling the tongue. So where will the child fall? It means the mother has to be heterozygous. Agreed? Yes, I'll be able to see the charts as we proceed. So we are talking about question number 4. Question number 4. Let me scroll down. Question number 4. Question number 4 says, create a Punnett square. Okay, thank you, Nyanchoka. You got it. Thank you so much for the response. Question number 4 says, create a Punnett square for a cross between homozygous purple flower plants and homozygous white flowering plant. This question is incomplete. Is it incomplete? No, it's not incomplete. P is still dominant. What is the phenotype and the genotype ratio of F1? Purple dominant. Okay, they've given us direction. Purple dominant over white. Still flower color. And here you can see the other question talks about a test cross on homozygous dominant, how they will appear. So I told you we have to start here, and don't get tired because that is your day. Just do it to your best. Whether it's exam day or the cut day, do it to your best. You don't get tired. Alleles, capital P for purple, small p for white. Then the first question says you draw a Punnett square for homozygous, meaning homozygous dominant, homozygous recessive. So purple, let's give the male, the female, purple, white, you know, we will repeat it until it will enter. You will get all heterozygous. And then, what is the question? That is Roman 1. They say, create a Punnett square. We've already created a Punnett square. It means we'll get marks for the trait, for the alleles, for the Punnett square. And now the question comes, the phenotype, no, the phenotypic and the genotypic ratio. Now they're talking about the genotypic and the phenotypic ratio. And you can see, here is where I say genotypic ratio. You can't talk about a ratio of 1, 1, 1, 1, or 4. Which ratio is that? All alike. Capital P, small p. Phenotypic ratio, all alike, purple. Yeah, that one is fine. Genotypic ratio, all alike, heterozygous. Phenotypic ratio, all alike, all will appear purple. Question number 2 says, still on the same, Roman 2, a test cross of homozygous dominant, a test cross. So for Margaret, a test cross is always done either homozygous dominant or homozygous recessive with the hybrid. So in this case, they are specifying that we use from here, any of the hybrids, remember, you still write male and female. Don't forget, I'm just trying to limit the space. Capital P, is it? Capital P, capital P, capital P, capital P, small p, capital P, small p. Did I get it right? Then you are asked, state the genotypic and the phenotypic ratio. You can't write the genotypic ratio all alike here. I said the possible genotypes are homozygous dominant, homozygous recessive, and heterozygous. So the genotypic ratio will come to 2 homozygous dominant to 2 heterozygous. And then you will say, for the phenotypic ratio, all purple. Because the law of dominance, the Mendelian law or principle of dominance, in heterozygous state, the dominant allele suppresses the recessive allele, so it's going to show purple. All purple. If you like all alike or all purple, it's still okay. But just by following the questions, you'll be able to get. So the questions can come using the plants, or we can dive into Mendelian disorders, and that's why I put there Mendelian disorders. We are on X-linked. And here at least I'm explaining the X-linked, the autosomal dominant, autosomal recessive. In exam, it's not easy for me to explain to you that it falls in which class, because that is what I'm trying to test. Because if I make it straight and say, oh, autosomal dominant, the student will know, yes, this is what I need to write in their leads. Yes. It will make it easier for the student to know how to do the Punnett square. So, X-linked, the numbering is continuous. In a family, and remember, I mentioned that for the X-linked, I will ask about the gender, who is affected. I have to ask about the gender, whether male or female, boy or a girl, because you have to include the alleles together with the sex chromosomes when you are defining the trait up there and the alleles. So in a family, father is hemophilic. This is what we did on Friday. Father is hemophilic. Let's start with the hemophilia, just by showing that we are working on hemophilia. Trait, and remember, it was X-linked recessive. That's what we saw. Trait, hemophilia. Give me the disease up there. Alleles, yes. Now let's read. Let's read what happens, the male, the female. Let us just put the X's ready. But then put small h. Present, that is what I said. It is X-linked recessive. The allele for the disease is recessive, but the disease affects the sex chromosomes. So you have to put for me these ones. Said Y is not touched. Only X is touched. Absent, the capital H. Let's go to our story. We see how it takes us. It says, in a family, a father is hemophilic. Father is hemophilic. The father, let's write the male, female, that one you can also write. So the father is hemophilic. And the mother while the mother is a carrier for hemophilia. We said the female is either a carrier, affected, or healthy. But the male, either affected or healthy. Those two options, because Y do not carry anything. Possible outcomes, then we can go to the questions. Possible outcomes. We have a female who is not affected, as much as a carrier is not affected. The allele for the disease is recessive, just the way it applies to the autosomal recessive disorders. A male who is healthy, a female who is hemophilic, and a male who is hemophilic. So the question says, state how many sons and daughters are infected and healthy. The daughters who are affected, one daughter affected, one daughter healthy, one boy healthy, one boy affected. Is the session recorded? Yes, it is going to record. Sorry, number 5. Question 4. I've skipped. We've skipped question 4. No. Margaret, I don't know which group, which book Margaret is reading. Did I skip? Somebody to tell me. Felix, let me answer Felix first. It will be question 5. Ah, good. Margaret, thank you. I'm going back there. I skipped question 5, correct. Felix, it's going to be recorded automatically. Immediately we are done. It takes like 25 minutes. And if you go to the live on my channel, if you go to the live sessions, you're going to see it. So you'll be able to revise pole pole at your free time. Thank you, everyone. Let me go back. Yeah, we've answered that one. So let me go back to question 5, kindly. Thank you for that. If I skip, let me know, please. Question 5, a monohybrid cross. A monohybrid cross on parental strains based on seed shape, round and wrinkled, as a trait generated, which were carriers of the trait. Kenya, I'll come back. Yes, that is where we are, the one which I skipped. Thank you. We are saying, that one you already know now, a monohybrid cross on parental strains based on seed shape as a trait generated F1s which were all carriers for the trait. Carriers. They came out as hybrids. They came out as heterozygous. It means, we are going to write it. Let's start. We have to start with the traits. We have to start with the traits very well. Nothing will be left unturned, no stone unturned. Seed shape. Give me trait here very well. Seed shape. Alleles. They've talked about the round one, correct. So the round one is dominant. The recessive, wrinkled. Simple. You'll get marks for that. But they are saying the parental generations generated carriers. It means we had pure breeds. Pure. We had pure breeds. We had pure breeds. If they're talking about carriers, they're talking about Heterozygous. It means the parental generations, parent 1, parent 2, they were pure breeds. So let's go back. What does it say? A monohybrid cross, one trait, seedship. Alleles, the dominant one takes the capital, the recessive one takes the lowercase, round wrinkled. They say a trait generated F1s, look at the F1s, which were all carriers for the trait. Use a Punnett square to conduct a test cross. You get? Use a Punnett square to conduct a test cross of the F1s. Punnett square. From the F1s, these are from the F1s, use a Punnett square to conduct a test cross with the F1s with the homozygous recessive. Remember here also, include female and male. Don't forget. What is the question asking? We go back to the question. State the genotypic and the phenotypic ratio of the F2s. Genotypic ratio. Genotypic ratio 2 to phenotypic ratio 2 round 2 wrinkled. Did you see it? Or should I project it? Are you seeing something? So they're talking much about the parental generations brought about the F1s, which were carriers, meaning from the pure breeds, we are going to get heterozygous. From the heterozygous, also still a test of the test cross, we specify that we are having homozygous recessive for the test cross. That one is fine. Just chat. When something is not clear, I'll be able to clarify. Don't worry. Question number 7. As much as number 6 was talking about hemophilia, let's look at 7 now. A hemophilic family, or now the man is the one who is healthy, involves a man who is healthy and his wife who is a carrier. Yes. Back again. We'll repeat until it enters. When you go to the exam room, you'll just be singing this song, singing. Because you'll remember Madame stressing on this point, and you'll be able to understand. So, the trait is hemophilia. The way I said, any of those diseases which you see there. Alleles. Remember, X-linked. Absent. What did I write? Absent. Present. Now they are saying that the male is what? This man is, the woman I could see was still a carrier, but now the man is what? The man is healthy. The man is free. Here the man is free. The man is free. The man is free. The man is free. Look at that. It says, the question says what? Draw a Punnett square to show the F1s. We've already drawn. State the gender. State the gender. And the number of kids who are healthy, that is without hemophilia allele. Without hemophilia allele. As much as the heterozygous one is also healthy, but they are talking about the ones who are not carriers. That is especially the lady, because the man cannot be a carrier. So the gender, which is male and female. There is a female without the allele. There is a male without the allele, which shows they are healthiest. Even the carrier one is healthy. But they are saying without the hemophilia allele. And it says again, state the gender and number of children who are carriers. A carrier can only be a girl. A boy cannot be. So I think even that question need to be revisited. It had to be reviewed, because it doesn't make sense. If they talk about carrier, yes, the man, the wife doesn't carry anything. So it's still, the gender is a female, and we have only one carrier. Just answer the way you've been asked. Which gender is hemophilic? Male. Simple as that. The Punnett square is going to answer. If you know, you know. You will sit comfortably waiting for Madam to start. When are we starting, Madam? Just say start. Question number 8. DMD, Duchenne muscular dystrophy. There is a group which was doing that. That is also X-linked recessive, where we are with hemophilia. DMD falls under that. Remember, we saw that the X-linked recessive are many, but then the X-linked dominant are fewer or rare. So most of the time which we gave, the examples which we gave for that table is for the X-linked recessive. Let us rub. If you have the energy, now the energy has to be there. You can write DMD. Maybe it is very long. Aldo, dystrophy, muscular, whatever. Come here. Put capital D, absent. Put small d, present. We are just using the same one for hemophilia, but now we are changing the letter. I will always give letters, even if here I might forget, but I always mention that we can use letter D, letter H, letter what? Yes. So let's read. This disease, where muscles become weaker over time. This one explains very nicely. I thought I'll leave it at that, and then the student will be able to figure out whether it is recessive. But let's finish the story. The gene is located on X chromosome. That already you should put the X, capital D, X small d. When you see it relates to the sex chromosome, that is X, put the X there. They are saying those afflicted with this disease usually die in childhood. If a female is a carrier for DMD, if a female is a carrier for DMD, marries who? Or who marries who? Or is married to who? Okay. If a female is a carrier of DMD, is married to a healthy male. Is married to a healthy male. Let's see the options here. Is married to a healthy male. Married to a healthy male. It was just like the hemophilia one. That is small d. I don't know whether it looks nice. I don't want the students not to get anything clear here. D. So they're asking, what percentage of her daughters will inherit the disease? What percentage of her daughters will inherit the disease percentage? Who will inherit the disease? It means the ones who are affected, the percentage. Not there. It is 0. Because in this case, however much it is a carrier, and they go and mate again with somebody who is free from the disease, it will still don't show any. So just as the law of dominance apply, it will still apply here. It will only be inherited if that daughter turns to be having the disease. In the case where the disease allele is recessive, it can't easily be transferred to the next generation. So you talk about 0. The question says what? Oh, it just wants the percentage of the daughters who can inherit the disease. There is no one who is sick there. And that one comes because in the male, the father is healthy. So as much as it's going to meet with the female who has the allele for the disease, but the healthiness in the father is going to suppress this one. So it is like it is not affected. If the female is affected, then it can easily be passed down the line to the other generations. The other one is colorblind. You know, colorblind also falls under the X-linked recessive. What percentage of his sons will be colorblind if a man is colorblind? Assuming the mother is not a carrier. Where is the daughter who is recessive? That is the boy. Nyanchoka. The boy is the one who is affected, the XY. The daughter is heterozygous, and the law of dominance says the dominant will suppress the recessive, so that daughter is not affected. When somebody is affected, they will easily pass to the next generation. So possibly it's the boy. If they could ask the question clearly that, where is it? That what percentage of her sons will inherit if the daughter doesn't show? It's a carrier, but the capital D is for the disease absent. So the possibility of the son inheriting more of the disease is higher than a carrier, because the law of dominance says the healthiness is going to suppress the recessive one for the disease. So for the autosomal recessive, for the X-linked recessive, when in carrier state, they are not affected. What percentage of our daughters? Oh no. I was talking about the colorblind. A man is colorblind. And colorblind also falls under the X-linked recessive. Somebody sees the color as orange. Somebody sees it as yellow. The other one sees it as luminous green. Now, the same one, they're still on X-linked. Color blind. Capital C absent. Small c present. They are saying the man is colorblind. It means now we change the man to be small. The woman, they are saying the woman is what? Is healthy. What percentage of the daughters? No. What percentage of his sons will be colorblind? Assuming the mother is not a carrier. The mother is not a carrier. It means the mother is totally healthy. Let's see the options. No one is sick. No one. Because the mother is healthy. No one is colorblind. What percentage of his sons will be colorblind? 0. 0%. Purely 0. Capital C. The mother being healthy. None of them. If she could be a carrier, then definitely one of the boys will turn out to be colorblind. But in this case, the mother is healthy. They've said the mother is not a carrier. Question 8. Question 8, we said it's also X-linked recessive. Mudoni, where didn't you understand? Can I take us, can we go back to the Punnett square? Or did you draw the Punnett square yourself, your side? They're explaining much about DMD, but in this case, they're saying the female is a carrier for DMD. We are going back to DMD now because we need to understand and we need to move the same pace. And I told you for these ones, write the disease there as a trait. DMD. And then we just leave these ones the way they are because they're still X-linked. This is also X-linked recessive. We are back to question 8 so that Mudoni can understand. They are explaining much about how DMD affects the muscles, become weaker over time, located on the X chromosome, so it's X-linked, recessive. The ones who are afflicted will die at childhood, still we follow the story. But then, if a female is a carrier, we are at DMD. Question number 8. Remember, absent, present. And the male is the male, healthy male, correct. Healthy male, capital D. We will have capital D, capital D, capital D, capital D, small d, small d. And why? Have we tackled 7 fully? Yes, we've tackled 7 fully. Mudoni, have we? Mudoni, why are you saying yes? Are you answering Malimu or we? Malim, are you answering Dennis? Oh, good. Dennis is answering the people up there. Thank you. If you help me, it's also fine. Mudoni, are you saying yes for the DMD? Because that one you already tackled, and I'm just trying to say here, the law of Mendel of dominance still applies. In the case of autosomal recessive, in the case of X-linked recessive, as long as the capital D is present with the small d, that person is not affected. So if not affected, it can't pass it down the line. Abdul is saying hi, hiing back. I'm hiing back. Yes. Mudoni, your yes shows that things are positive. I want us to move at the same pace. And I always don't like to talk too much until the students get bored. Thank you. So, if we reach a point where you think the brain is saturated, I will stop. Because me, I can talk until I tackle all of them. But it is not like the world is ending today. Are you tackling so that you can finish the questions, or did the students understand? You get the point. So we already tackled question number 9. I want us to move on the same page. Nobody is left behind, because we are here to understand. Oh, good. We are at autosomal dominant. Correct. I think we are moving on well. Question number 10. It specifically now states autosomal dominant. I just give a few examples so that we can know how to show our alleles, how to show our traits, how to draw the Punnett square. So, for the autosomal dominant disorders, I told you that the allele for the disease is dominant. And they talked about expressivity varies in individuals. And it also talked about, still in heterozygous state, it will be expressed. So you can see how grave it is. The allele for the disease is dominant. Let's see. A heterozygous male for achondroplasia. The trait up here. Now let's rub. The trait up here is achondroplasia. Achondroplasia. Alleles. Here, don't put for me the sex chromosomes kindly, because here I'll ask about the children who are affected and the ones who are not affected. I will not ask about the gender because it doesn't touch the sex chromosome. So you say capital A present, small a absent. Come to the Punnett square. Now let's proceed with the story. A heterozygous male mates with an affected female. Heterozygous male. So you get capital A, small a. Female will be small a, such that, let us see the options. Let's go to the question now. I hope the students can see. Okay. Good. I can see some has answered what I kept on asking. So, using A, list the number of affected and unaffected children. Affected are 2. Unaffected are 2. Simple as that. The Punnett square is going to help us to solve all the problems, genetic problems, sorry. So the allele, I'll look at those alleles. Just by looking at, I'm not going to explain that it is autosomal dominant and capital A is, I just put achondroplasia there. I just put neurofibromatosis there. I will just put progeria there. I will just put maybe the sickle cell there. I will just put the phenylketonuria there, so that the students will be able to identify which class of the Mendelian disorders does the disease fall under. Then after that, I'll see the Punnett square. Yes, very interesting. So we are talking about progeria, Another example, progeria. So we just rub and we put hours, capital P, it is present. Small p, it's absent. Then the question comes, an affected male for progeria mates with an affected female. Ah, how is this question? No, it doesn't make sense. How can they both be unaffected? Then there is nothing we're asking. Progeria is not there. Okay, I think it was supposed to be affected male. And it has to be affected maybe in heterozygous, which I have to mention, or homozygous recessive. Let's assume the male is affected in heterozygous. The question was a bit awkward. That is not what I meant. The female is unaffected because both of them cannot be unaffected. Then why are we looking at the disease? Then none of them is sick. So in the case where the male is affected but heterozygous or a carrier, you'll have, they talk about what are the chances. You'll have 50-50 affected children and 50 affected, 50% unaffected. What are the chances of a child having the disease? If both, okay, if both of the parents are, okay, now I get it. Unaffected because they were both heterozygous. So let's revisit. Let's revisit capital P, small p, small p, small p. So it explains much the genotypes of the parents at the time now when square. But now they talk about both parents are unaffected. It means they are both heterozygous or carriers, but then the allele for the disease is dominant. So it should talk about the unaffected, meaning the affected, not the unaffected. Because progeria is autosomal. Did I mix them? No, I didn't mix. Was I reading number? I was reading number 13. Oh no. Sorry. Oh, that is question number 13. Yes, that's what I've realized. I'm reading a wrong question. But you can see the question number 11 was not making, it wasn't complete. So the unaffected male for progeria mates with an affected female, that one doesn't match. Then why are we looking at the disease? So I already assumed we can use the first one where the male is heterozygous, the female is homozygous recessive. So just the way the genotypes are expressed. And if you see a question like this, number 11, in the cat or the main exam, raise up your hand because that is an error. It has to be corrected, because if it's not corrected, you will take much time trying to scratch your head, thinking that you are the one who is not getting the question right. But in real sense, that question, I didn't write it correctly. Because if both of them are unaffected, then it doesn't make sense. I don't know why the light is coming to something color which I don't know, but I assume the students can see. Can you people see? Oh, it's one of my lights which has ran out. I hope you can still see. Yeah. So we are moving to question number 12. Thank you, Sam. Thank you. We are moving to question number 12. Because question number 11, it ought to be one of the parents has to be affected. Akinyi Miriam, hi. Thank you. Thank you if you can see. Thank you so much. Now, we are talking about one of the parents has to be affected. So question 11, I had set it wrongly. So that's why I was saying, if you see such a question, raise up your hand, because it can't make sense. It can't make sense if both of them are unaffected and the disease allele is dominant. So you'll know how the scenario goes so that we can have a Punnett square which we can understand. Question number 12. Thank you so much. If you people can see. A woman heterozygous for sickle cell anemia. We are now at autosomal recessive disorders. We've done the chromosomes. So you know the autosomes, you know the sex chromosomes. So we are still on autosomes. And we are on sickle cell anemia. The capital S, absent, disease is not there. Small s, disease present. Let's start from there. Then the story goes that a woman heterozygous for sickle cell anemia mates with a heterozygous male. Female and male, both heterozygous. Female and male, both heterozygous. It means capital S, small s, capital S, small s. The affected will be 25% chance of getting affected child. So let's see. List the number of affected and unaffected children. And you can see the pattern. When we are talking about autosomal disorders, I will not touch on the gender. The gender is only determined by the sex chromosomes. So that's why you can see I look at the probability of the of the affected child, the number of affected children, the ratio of affected to unaffected, something like that. So we are saying list the number of affected and unaffected. Unaffected are 3. Affected is 1, right? Because the law of dominance will prevail. The capital S will suppress the recessive, and then those 3 will appear healthy. That's what we saw. Cystic fibrosis, still in the same class. So let us change sickle cell with cystic. Cystic fibrosis. Still, use capital C, absent, small c, present. The question says what? So they talk much about it's caused by recessive allele on chromosome 7. A person must inherit 2 of these alleles to have the disease. It means when you are homozygous recessive, that is when you are affected. Using C, you can see they're talking about a person must inherit the 2 of these alleles to have the disease. So that's why the DMD, I was talking about the carrier, that girl cannot spread the disease to the next generation because the law of dominance will apply. So a person must inherit 2 of these, Using C, what are the chances of a child having the disease if both parents are heterozygous? Because the characteristics were reading very well that there is 25% chance of getting a child who is affected in the autosomal recessive disorders. So we are talking much about 1 out of 4. 1 out of 4. So they talk about, the question says chances, that is probability, percentage. 25%, right? 25% chance. We are moving to dihybrid cross. The independent assortment. The law of independent assortment. As much as those genes control those two traits, but they don't depend on one another at the point of segregation. The gene for shape segregates independently from the gene for color, for example. The dihybrid cross will be able to explain much about this, and I really love this one. I think the question number 19 is repeated. We'll finish. Don't worry, we'll finish. Just be patient, then you'll just go and revise comfortably at your free time until Friday noon. Good. Dihybrid course. You know the reason why I'm saying we are almost, because even by revising the classification of genes, that is something somebody can understand, can read and understand and know what to write, because there were several. Classification of chromosomes based on the position of the centromere. That is something which is easy to understand. You can just draw, contrary to these ones. So in dihybrid cross, we talked much about this cross is the one which explains much of the independent assortment principle. Let's go straight to the question. Gene A controls fin length in this complicated name of a fish, Plecostomus. Where it got it from, I don't know. Short fins are dominant to long fins. Gene B controls the fin color, gray and lemon. Gray dominant over lemon. Imagine this question shows very clearly letter A and B. Somebody will come with G. They come with G and S. So you wonder, where does the G and the S come from? Why don't you represent the fin length with A? So the dominant one will get capital A. Let me explain as on the board as I talk or as I speak. So you talk about two traits, dihybrid, two traits. We have fin trait. Fin what? Fin length. Fin length. Here at fin length, finish, capital A, small a. We are talking about fin length. We have short fins are dominant to long fins. Talk about short, long. Trait, if you like, do trait 2. Fin color, I think. Fin color. Is it still talking about the fins? Let me see. Fin color, gray, dominant over lemon. Capital B, gray. Small b, lemon. Put it clearly. Punnett square for question 14 should be more than 4 marks. Correct. I'll adjust the marks because Some of the marks do not apply. They are a bit less. You get? Don't worry about the marks. What marks did I do for the Punnett square? Oh, we are in that question. This question always takes 10 marks. So I always spread it out. But the Punnett square will have much of the marks. Because, you know, you have to work with the gametes, combinations, draw a Punnett square. So I always put it, everything, 10 marks. So I know how to spread out. So don't worry. Some don't worry about the marks. Let's just get the concept. That one will be adjusted so that you don't struggle so much and then you like, oh, see, 4 marks. It's always total to 10 based on the question which is asked. So we are talking about, let's work with this. We generate the gametes, we do the Punnett square, then we can go straight to the question and know what to answer. So I said in this case, you have to generate the gametes. Remember, the formula for generation of gametes was based on the number of heterozygous, 2 to power n. In this case, we have 2 of the heterozygous, means 2 times 2. You will get 4 of the gametes. So, talk about getting the gametes. This goes with this. This goes with this. This goes with this. And this with this. Same for the males and females. Then do a Punnett square. Simple as that. Do a Punnett square. Put the gametes. Then it will come to 9, 3, 3, 1. Same, that is male. Female here. AB. A capital small b. Small a capital B. A small b small. So you will have AA, BB, AA, BB, which is heterozygous, heterozygous, capital B, capital B, capital A, small a, capital B, small b, capital A, capital B, small b, capital A, small b, capital A, small a, capital B, small b, capital A, small a, small b, capital A, small a, capital B, small b, capital B, sorry, capital A, small a, capital B, small b, small a, capital B, small a, capital B, small b. Did I get it right? Capital A, small a, capital B, small b. Capital A, small b, small b. Small a, capital B, small b. Small a, small b, small b. Such that we should confirm that we have 9 short gray, 3 short lemon, 3 long gray, and lastly 1 long lemon. Let's see. Short gray, they have to be 9. We'll do a tick. 1, 2, 3, 4, 5, 6, 7, 8, 9. We'll do for short and lemon and X short and lemon here. 1, 2, 3. We'll do for long and gray, we'll do a Z. 3, 2, sorry, 3. And then this one is 1. So it matches very, very well. Yes, I'll move it. I just wanted to finish. I'll move it very well. Look at the gametes which you've generated. Put them well. And you know, this one applies to all in dihybrid. These questions in dihybrid which you see, it is going to apply to all. We are looking at 2 traits. The genes which are controlling those 2 traits will have to be independent at the point of segregation. They will independently assort. The gene which controls fin length do not depend on the gene which controls the color. We are talking about fin length and the fin color. So first, mention the traits, each one of them, the alleles, each one of them. Based on the letter which is given, generate the gametes. Because already dihybrid, you will have 4 gametes now, because you have 2 heterozygous. Those gametes, take them well up there in the Punnett square. Because even question 15, you also have 2 traits. Show the possible alleles for the experiment, just as whatever we've seen in 14. State the gamete combinations, what I've done down here. If you know the 4 alleles, definitely go straight and do the cross, which I've done there. You generate the gametes. Draw a Punnett square. From the gametes, you do a Punnett square. Clearly show the phenotypes of F1. You know, the phenotypes, you'll talk about 9 short gray, 3 short lemon, short gray, short lemon, long gray, long lemon. And then you'll come to the phenotypic ratio. You'll say 9 short gray, colon 3 short lemon, colon 3 long gray, common 1 long lemon. So it's not difficult. Even question number 16 talks about the checkered. The story can be long. So don't be confused in between, but then focus straight on the alleles. We are talking about 2 traits, 2 genes, independently assorting. So kindly put the alleles clearly, and then next, generate the gametes. After generating the gametes, go straight and put a Punnett square. So that's how the dihybrid works. For all which you are seeing here. Am I repeating myself? Oh, that is monohybrid, sorry. Yeah. If you go through until question number 18 is the same. Just identify those traits. After identifying the traits, the alleles, generate the gametes. After generation of the gametes, do a Punnett square. There is a question which talks about the DMD is the same. There is one with the rickets. This one I just added when I was almost. Kindly number 16. In pigeons, a dominant allele C. Checkered pattern. You talk about feather pattern. Feather pattern. So if I direct you to whatever we are going to do, definitely at that point, you'll be able to know how to generate the gametes. Feather pattern. Then you talk about which one is dominant. C, checkered pattern in feathers, it's recessive allele. C, okay. Dominant allele C for checkered. Small for plain. Feather coloration is controlled by an independently assorting gene. A dominant allele B, feather color. Feather color. Brown and red. Here B comes for red, for brown, for red. Then brown, or did I put it the other way around? Let me see. Feather color, feather pattern. B produces red feathers, yes, capital B. Recessive B produces brown feathers. Birds from a true breeding checkered red variety are crossed to birds from true breeding plain. Is it the true breeding? Generate gametes and the crosses. Repeat how to generate the gametes. That is why I'm doing this one, Malim. So, they are talking about true breed. That is where the confusion will come in. If you talk about the true breed, in each of the statements we've been looking at in the monohybrid, you will see that the result will be a hybrid. And in that hybrid is where you can see all their leaves are represented there. So, in this case, leave alone the true breed, where they say the true breed, whatever we are seeing here is, if you have true breed for the checkered, plain and checkered, you are going to get a hybrid. A hybrid means we are representing the dominant allele and the recessive allele. It is heterozygous. So that is why, aside of all the true breed, crossed, and then you get whatever, go straight, write the alleles the way I've written. If you already have this and you got it right, from the dominant one, the recessive one, the two genes which now control the pattern of the feather and the color, Malim, this is how we are going to generate the gametes. That is how you are going to generate the gametes. If you generate those gametes, line them up there at the Punnett square, like a matrix, the way you are doing a matrix sum. If you see the dihybrid, if you do a pure cross for the checkered and plain, the options you'll get there is a hybrid of capital C, small c. You do a pure breed of feather color, red and brown, pure breed. We are number 16 because the formula applies the same until number 18. So somebody requested for me to do number 16, and that's what I'm showing until you get the gametes down here. Malim was asking how to generate the gametes. Down here you can see the gametes. Can you see? So you'll always have to generate the gametes. And since there'll be 2 of the heterozygous, because from the pure breeds, you'll get a hybrid for the feather pattern. You'll get the hybrid for the feather color. Now, from those hybrids, the pure breeds you're talking much about, you have to generate the gametes there. And that's why you have to explain each of the alleles up there. Then you'll generate the gametes. I've done the crosses. After generating the gametes, arrange them up there for the male and the female. Then do the matrix there. You will get the 9, 3, 3, 1. So you will get 9 checkered red, 3 checkered brown, 3 red plain, and 1 plain brown. That's how it goes. So it applies to all, which you can see in dihybrid, just by getting the point that we have 2 genes which control a trait. Those 2 genes have 2 alleles. Each of them has 2 alleles. The alternate form of those genes, the contrasting characteristics, whenever you see a physical expression or a phenotype. So after identifying those alleles, we go straight and generate the gametes, and then after that, fix it on a Punnett square. I wanted to highlight on rickettsia. You know, rickettsia is sex-linked dominant. The only one which we saw, which we could discuss in class. The vitamin D resistant, something like that. So, whatever you have to do, the way we did it in class, since it is X-linked, the X has to be there. The X chromosomes. And then capital R present. Small r absent. Then you do the Punnett square, the way you can understand the question. Are we together? The other one talks much about, so the man has rickets. It means the man is X capital R Y. And the female is X small r, because they're saying the woman is not a carrier. So just the way we worked on hemophilia, the way we worked on colorblind, the way we worked on DMD, but now the difference here is we are going to swap the alleles for the disease. In the case of rickettsia, the allele for the disease is dominant. In the case of hemophilia, DMD, the allele for the disease is recessive. Just by understanding the simple concept, it will be very easier for you to understand. So it talks about question number 21. Classification on chromosomes based on the position of the centromere. That is simple. And it can appear anywhere. If I don't want the students to go without marks, maybe we also have to add, I don't know why, I think it's almost, my battery is almost going off, but I'm done. So you also have to add, at the point we're talking about the position of the centromere, we are talking about the metacentric, acrocentric, telocentric, submetacentric. And then you have to describe the position of the centromere and the shape, how we can see it in anaphase. So you have to explain the position of the centromere. So when you are told about submetacentric, and then you talk about submedian, what is submedian? It's up above the median part, the middle part. Metacentric, it's median. What is median? It's at the center. Acrocentric, almost to the end. And they talk about the small arm exists, but it's difficult to see it, though it's still visible. And then telocentric at the end, the telomeres. That's how simple. And then you draw the structures and you'll get the marks. But nowadays I see 20 marks is too much. Oh yes, I do 10. 10 is fine. Even if I give you half, but 10 is enough. So the other ones just talk about submetacentric and metacentric. You know, the sub is up the middle point. Metacentric is at the center. The centromere is at the center. Telocentric at the end. Classification of genes. We had several. Pleiotropic, lethal, basic. Define any of those, 7 of them. You'll get marks. So you have to mention and define. That's fine for today. Thank you so much for turning up. So I wish you well as we are going to revise. I hope I've been of help to somebody who could not understand something which was not clear. So for now, you know, maybe the eye has been opened a little bit. Welcome. Thank you so much. The eye has been opened a little bit. We only have a few weeks. So let us just keep the effort. Keep the fire burning and it shall be well. You know, our exam, we are doing it on 14th. I saw it on 14th. So it's that soon. But I know we are going to do well. The effort which you've put cannot go unpaid. Just sacrifice. Keep on moving. Don't wait until the last day, then you take cold water and a trough and you put your feet. My friend, you'll be wasting your time. So thank you so much. Good night and have a lovely week ahead. God bless.